Alright, macromolecules. This may have to be 2 videos, but we're going to try really hard to do it in one. Most macromolecules are polymers. A polymer is a large molecule made up of identical or similar subunits hooked together. So you can see right here, here's your monomer, your subunit. Monomer, polymer, right? And then it just repeats and repeats and repeats and repeats. And most of these guys are going to work that way. A macromolecule, macro means big, is a large organic polymer, and we're going to talk about 4 types: carbohydrates, lipids, proteins, and nucleic acids. And that's what the following slides will be about. A polymerization reaction, and I didn't put that on here. Let me A polymerization reaction is a chemical reaction that links 2 or more small molecules to form larger molecules with repeating structural subunits. We're going to talk more about condensation reactions. We'll see this term a lot more. Condensation reactions are where monomers are covalently linked by the removal of a water molecule. One monomer loses an OH, the other one loses an H. This requires energy. It requires catalysts or enzymes. But this condensation reaction, let me pause. Alright. The condensation reaction removes water to build molecules, and hydrolysis adds water to break apart molecules. 40 to 50 monomers are used to construct almost everything. New properties emerge when they're arranged in different ways. That's the unity. Alright, so up first is carbohydrates. Carbohydrates are organic molecules made up of sugar and sugar polymers. If we see the monomer is a monosaccharide, this just means it's made up of one sugar. These are simple sugars, things like glucose and fructose over here. Simple, right? They have this is actually the chemical formula or molecular formula for glucose and fructose. They are isomers. There's always 2 times as many hydrogen as the carbon and oxygen. Carbon and oxygen are equal for monomers. These are major nutrients for the cells. Glucose is the most common. It's produced by photosynthesis. The carbon skeletons are raw material for other organic molecules. The energy in all those chemical bonds is harvested in cellular respiration. Characteristics of a sugar. The carbon skeletons vary from 3 to 7 carbons. The spatial arrangement is asymmetric. And in aqueous solutions, many of them form rings. And we'll see glucose here. Each of these little rings is glucose. There's also an OH group attached to each carbon except for one that has a double bond, and you can see that here and here, so that's there. Disaccharides, di is going to mean 2. These are going to be things like maltose, lactose, and sucrose. Maltose is 2 glucoses combined. Lactose is glucose plus galactose. It's found in milk. And then glucose and fructose is sucrose, and that's table sugar. Polysaccharides have lots and lots and lots of monomers linked together. Here's 3 of them over here. You can have usually a few hundred to even thousands of simple sugars making up these guys. You have 2 important functions, storage and structure. And with storage, we have 2, starch and glycogen. And starch is how plants store sugar, and glycogen is how animals store sugar. Let's see. In plants, starch is usually stored as granules and plastids, and in animals, glycogen is stored, or in vertebrates, glycogen is stored in muscles and the liver. For structure, we have cellulose. I'm going to do that in a different color, sorry. Cellulose and chitin. Cellulose is the major component of plant cell walls. It cannot be digested by most organisms, including humans, and it's still important in our diet as fiber. A lot of critters, like cows and termites, have symbiotic bacteria or flagellates that help them break down the cellulose to get more nutrients out of that. Chitin is what forms the exoskeletons of arthropods and is found in cell walls of some fungi. Lipids are a very diverse group. There are only one that's not a true polymer, but they are linked together because they are insoluble in water. And we have 3 important groups here. We have fats, and you can see fats are made up of a glycerol and a fatty acid. And that's here a triglycerol. They vary in number and location of double bonds. You can get different types. If there's no double bonds between carbons, you have a saturated fat. These are found in animal fats. They're solid in room temperature, and it's thought that they can line up together more efficiently to clog arteries. So these are bad guys, kind of. If you have a double bond, like right here, this is an unsaturated fat. They are found in plant fats, most plant fats, and they are liquid at room temperature. You have something called a trans fat. And trans fats are unsaturated fats that are artificially hydrogenated. They add hydrogens to it so that it's solid at room temperature, and these guys are bad. And that is, functions of fat. It's compact and lightweight compared to starch, and since animals have to move around and be able to carry fat, it's more efficient for storage in animals. It cushions organs of mammals, insulates against heat loss. Phospholipids, these guys right here are phospholipids. They make up your cell membranes. It is a glycerol and 2 fatty acids and a phosphate group. So you can see glycerol, phosphate, choline, fatty acids. They cluster if they're put in water, they cluster to where the hydrophobic ends, the fatty acid tails, hydrophobic, are here in the middle together, and the hydrophilic are towards the outside. Again, cell membranes, phospholipid bilayer. Steroids are lipids with 4 fused carbon rings with various functional groups attached. Main example is cholesterol. It's an important compound of animal cell membranes. It's a precursor to other steroids like vertebrate sex hormones and bile acids, and it can contribute to arteriosclerosis. I think we're going to stop this video now, and I'll do the proteins and nucleic acids on another video. So this is the end.